probably the most. UK Biobank, I don't feel very patriotic about the UK because it's very expensive. So UK, I mean, if you want to use it, I mean, it's very expensive. But that's the biggest data set. So it's not the most curated, the biggest data set, so the deep learning is very good. So this first question is data quality, and if you, as a student, want a data set, I mean, it'd be annoying to spend, I think, Hongtu, as you mentioned, a year sorting out mistakes and missing data and confusion. So I notice when people do challenges at MICI, they might say, hey, run your algorithm on these 3 data sets. And during the challenge, the competitors say, hey, did you notice there's some problem with the data set? So I think the first solution is make the data public and have as many people as possible help curate it. I don't know, others may have good ideas. I mean, maybe they QC it and upload the QC stuff back to the original database. So that's the first thing. Second one is data sharing. And we all did a lot of data sharing. I think for Alzheimer's, there is good public data. And I don't know why that. Maybe it's very well funded. A lot of people are working on Alzheimer's and it improved. For other diseases, there's less shared data. I mean, many of you worked on, I think you worked on FBIR, schizophrenia. There's some schizophrenia data. Other disorders, less public data. There's some in oncology, there's some public tumor data. I feel like, I mean, if you want, I mean, Enigma is an international network with data. You can get data from any disease. I mean, just ask for getting data from any disease. There's one privacy issue that keeps coming up though. So let's say someone has a lot of data on HIV in the brain, and that's sensitive. I mean, someone might take part in a study, they're HIV positive, and they're concerned about the data sharing. And maybe that happens with other diseases. I just picked in HIV. So to protect the people whose data it is, there's all this bureaucracy of data sharing agreements, material transfer agreements, IRB. I mean, it's good. I mean, you would want there to be safeguards. But for a computer scientist entering the field, I mean, a lot of people say, well, hey, I kind of like ImageNet because I could download it, or I like Acme because I can download it. There's not a whole lot of messing around. So what I've noticed is that the barrier to entry for lots of disease data sets, in theory, can be surmounted, but in practice, people give up. I mean, it's just too much work. Nobody has enough time or hours in the day to do it. So most AI students, I mean, students who are going to work with us on AI, will pick a very well curated, easy to get data set. Maybe the winner is ADNI or ABC. I don't know, each of us likes certain data sets. But I think if we want to kind of fix this or improve it or something like that, you got to work from both sides. Either curate some data sets and make them simpler to work with without errors. Or I don't know the answer to the other one. I mean, the privacy advocates would say, well, I don't really think UK Biobank should be public. And you'll have seen in recent weeks, people that downloaded UK Biobank are now being told to delete their data and only use the server in the UK. And I mean, I have an opinion on that. but I mean, they're not doing it just for a laugh. I mean, they're doing it because they're worried about privacy, and in the UK and in Europe generally, there's very harsh data protection laws about privacy, so they don't want to risk their project's reputation by having people, do other things. So that was sort of a long version of what Dar said.
So, yeah, I completely agree with the points made before. Actually, I think the question has been basically answered now. But I will only add that maybe something else to be focusing on is also some basic level of preprocessing done post data collection. I wonder how many labs have reprocessed the ADNI data on their own, and this feels a bit of a waste of resources, right? And also doing some basic level of reprocessing also allows to check quality control and deal with that issue. Also, the issue is that once a lab decides to do the data reprocessing on their own, then of course they once invested a lot of resources, they are not super keen to share the data at that point. So there's this whole dynamic of people sticking to their data, not knowing how the reprocessing was really done. So the only point I would add, it would be great to do some level of processing. I think I really like what ACP did and how quickly they caught up in terms of how used the data set is as a benchmark, exactly because there was some sort of minimum processing done to the data.
I think this is a very good question. I think a majority of the the audience are working on data sciences, including myself. But we usually overlook the quality of the data, the source of the data. This is a lesson I learned in the last 8 years when collaborating with Vince, Julia, Stephen, and Tony Wilson from the 3 other states, and we have a large consortium to study the breed development, and we have 2 data collection sets in New Mexico and also in Nebraska, and so at the beginning we do very rigorous quality control and make sure we follow the same protocol. Even though we use different scanners, we wanted to do data normalization, and also you have a lot of experience on the data harmonization, right? Make sure we get rid of the bias, and for the data pre-processing, I think it's a very important step. Even though we focus on model development, algorithm design. So we have to understand the quality of the data, how the data are generated from the beginning. And for the students, in my lab, I usually let them to work on pre-processing the data. So I got a pipeline from Vince Cajon and also others, and I let them to run the pipeline to do data smoothing, noise removal, normalization. So this way they can understand how the data are generated, how the variation in the data can cause a difference in the subsequent processing. So it's a 10 years time-consuming work, and but we have to do it. And similarly, for genomic data, we do data imputation and data normalization. Because there are a lot of missing data in the samples, and also it's hard to control the data quality when collecting the samples. We have a large center for genomics, especially multi-omic study, and we encourage the students to go to the on-site clinical recruitment place, and to monitor how the data are collected. So this way we have a good sense of data curation, data management. I think now UNC is implementing a more rigorous data management and sharing plan, which is very good. And data is a treasure. Before we do machine learning, before we do statistical modeling, we need to have a good quality of data to integrate the multiple source of the data integration to get rid of the variation. And eventually, we have to do data validation, right? We go back to the data source and test the data from different labs, from different platforms, to do external validation to make sure the funding can be consistent. That's what we do.
Yes, so I will just be quick. I think majority of the point has been covered, and I agree also effort should be put in the processing part. My experience, particularly for lots of processing of the, for instance, functional connectivity, which our group has been working a lot, and I have a bunch of students working with different collaborators from different institutes, and they actually may use different pipelines. Even for the same data set, we got eventually different sample size after we hope to get the same functional connectivity. I think the harmonization of the processing pipeline is super important. And on the other hand, I feel also part of the data quality, or even the data we actually get, sometimes we cannot control. But as a data scientist, whether we can try to bring some of the methods, even some innovations to try to use analytical tool to at least within the bandwidth to improve the result we get. For instance, we know for Alzheimer's, lots of us are working on the preclinical stage is the silent part. We don't have symptoms. So typically there's not a lot of observation for that, or whether we can use a more just advanced analytical model try to still uncover what borrow information from the subjects to get what it looks like, or using more higher quality data like HCP data to improve the quality of other just lower quality like ABC imaging stuff.
Thanks. But I think one emphasize, I think you cover most of the question points I want to cover. But I think I'm most concerned is about imaging processing components. You run through A from A to D, right? Like you can make sure that basically at the beginning data quality acquisition is good, right? That's the first starting point. Answer the right questions, you get the right subjects, then get the data. Imaging. The next question is for all the imaging processing components. Do we have any kinds of existing data to validate all these algorithms? I think the answer is no, in general, right? I think it's just the segmentation. If you're looking for subcortical regions, we don't know what segmentation algorithms we're using is function, correct, right? And whether the algorithm is robust to all kinds of different data sets we're using, right? That's the I think that's because all the downstream analysis we're doing, if it depends on segmentation or imaging reconstruction and the retration, how much will we trust that? How much will that influence? Now, even though sometimes, for deep learning method, for most of deep learning methods, we don't think that retration is that critically important these days, right? And how much we can trust all these imaging preprocessing components, the effects of these imaging preprocessors on the downstream analysis, like you can do genomics, other things, right? Classification, all the things. What's your opinion on this? I raised the questions because I have been working on over 20 years. I just feel that we need to create the community, we need to create this kind of data to validate all these major pre-processing pipelines from a rigorous perspective. I think that you think about the AI or deep learning, this stuff come out because the FIFI really spent tons of effort on creating the data sets for that purposes. But as a community, your imaging community, we have been working on for years, right? Do we have this kind of data set or resources, or effort to devote it to that kind of questions and basically move this field forward? Because that's very important in general. Even you're doing genomics, omics data, that's some questions come up. Why should we trust this? Now that's the basically that's my key questions in my heart, I think that's the something we can do as a community, because that's our job. I know this is very tedious, and sometimes there's no ground truth behind it. But we can do something on that. That's the kind of my comments. Okay, I think that's tough. And I move to the next one because the time is short. And the exciting areas of research emerging from imaging genetics. Okay, I think because in particular I designed the question for Tom, for Paul. Looking forward, what are the most exciting areas of research you believe will emerge from the action of the imaging genetics from the methodological and the applied perspective in one sense? You think about it, you have been done this for a while, and see that basically because you have connected this larger number of subjects with both multiple data types. And I think that in the future, as the AI invest a ton of resources on doing the similar things, And as you as a pioneer in the field, what's your opinion on that?
Yeah, I'll have a go.
Yes,
I'll have a go. So this is a great question. And our center's called the Imaging Genetics Center, so we should probably have a good answer to this. So I have a clinical answer and an intellectual answer. This is where a lot of us are mathematicians. So the clinical answer would come from NIH a little bit like this. They'd say a lot of the major diseases that affect people, Alzheimer's, Parkinson's, cancer, there's a genomic driver of the disease, and we want to interfere with it. And so in the case of Alzheimer's, I mean, maybe amyloid is building up in the brain, tau is building up in the brain. This is a very clinical answer. And the geneticists at NIH say, if we can have a better understanding of the genetic driver of a disease, not just the gene sequence, but the omics and the pathway, you can do genome-guided drug discovery. And there's a lot of grants like this where they say, well, this particular gene looks implicated, it has a strong effect. If we can interfere with it either at the RNA level, at the transcriptome level, or at the protein level with a vaccine, right? These are all ways drug companies can block a disease. I mean, amyloid plaques build up in the brain of vaccines. There's the amyloid plaques. People, they don't get better, but they get worse, slower. Okay, so that's the NIH answer. They say, could you please direct your effort towards finding genomic and omics pathways that drug developers can interfere with and block? And you need information to be able to design these pathways. So in psychiatry, you go to Janssen, or you might go to Biogen, and they say, in schizophrenia or in major depression, we're still trying to develop drugs that counteract omics and genomics pathways, and we don't fully know what they are. Could you please go and look for what these pathways are, and we can do genome-guided drug discovery. So that's a sort of commercial, industrial NIH perspective. It's like, how would you stop the disease? But then there's also an intellectual answer. So many of you are mathematicians, and you like problems. I mean, you might like problems because you can solve a practical problem in the world. Or you might like a problem because it's a really interesting intellectual challenge, like a pure mathematical. And I think connecting imaging genetics, we saw cool methods from UPenn with the joint DSPARS CCA, and there's been other presentations. I think you were talking about two modalities of Bayesian methods to reconstruct connections between them. I think it's a really interesting mathematical question. How do you connect two data domains? I mean, you've seen text being connected to video in the last 5-10 years, which we think is amazing. I mean, how does cross-attention learn those matches? So I think for those of you that are pure mathematicians or on the pure end of computer science, I mean, that's just an unbelievably cool intellectual challenge. I mean, it's sort of like Fermat's last theorem, or if you're interested in sort of unsolved or very difficult problems. And to me, that's another reason to do it. So it's a little bit of a philosophical reason to do it, like why do we do the things we do? You could do it because you want to find the practical solution to a disease in the best possible way with the most intelligent scientists to do it. That's the NIH answer. But I think many of us went into mathematics and computer science because we like the intellectual challenge. And luckily, the people that are the best at the intellectual challenge are usually pretty good at the practical challenge as well. So that's two answers to your question.
I think probably you've pretty much got the sort of. I actually have the questions and a lot from your answers, because we are just imaging scientists and we are learning genomics, and through the genomics work, the initial work we are doing, we found actually linking to the previous question about the data availability. Initially, we started with SNP data and the image data, which is the source of the disease. Everything starts from biology, and image data is the end of phenotypes point. And by linking that, we are actually assuming a lot of modeling assumptions, because everything that happens in between is lifestyle change and comorbidities and health education and sort of human health, and all those things are not part of the data. And I then realized, okay, so probably we need to have the multi-omics data to connect all these dots and see what's the factors affecting those. And then the problem itself become more complicated because you need to have data. You have more linked resources to do that. But I think that's the that's the sparsity of the data of resources is the next frontier. Something we will have to have this multi-layer data, and I see NIH is creating a multi-level, a multi-scale it's emphasized on the multi-scale modeling, pretty much connecting all those. I think probably it's the next generations of science, or previous generations of scientists need to generate those data in order for the next generation to solve the problems. I guess it might be varying depending on field to field. Like in what we're doing on brain, I feel it's the most challenging because a lot of times we cannot have the direct home data from the brains because of brain barrier, and also the clinical. It might be easier for cancer, but then I think eventually the solving the issue needs the data scientists working with like a data, the basic scientists to collect the data and working together. That's my 2 cents.
Thanks. So yeah, another interesting aspect of imaging genetics, I feel like, is I was actually recently talking to someone about this, and was like, how's that no one has ever applied a large language model to SNPs data and trying to predict a trait of interest, trying a complex trait? It was like, no, the reason you don't see publications out there is because there's no positive results. There's a lot of people who are trying hard to apply these models to this type of data. It's just that doesn't seem to work. So I feel like this is interesting actually, because maybe what's specific about genetics data is a lot of irrelevant information there, maybe a lot of redundant information. So maybe a lot of these sophisticated models that work very well in capturing complicated relationship don't work as well. I mean, when we think of the typical applications of these AI models, these are not settings where there's a lot of noise, and no setting where there's a lot of redundant information. It's more about capturing very complicated relationship. So I feel like this probably imaging genetics falls in a setting where there might be complicated relationship between genetics and the imaging traits that we see, but this is also a noisy setting with a lot of redundant information. So really it goes back maybe to the statistical foundation notions of bias variance trade-off, and that's why I think this is actually a good opportunity to work on this.
Well, this is really a good question. I always ask myself, working on imaging genomics, what should we do, right? Yeah, and I talked to the UNC-EPIC program officer, recently, and it looks like they are not quite interested in imaging genomics. And in May there was also a special session at the EPIC dedicated to imaging genomics. And for us, we mostly focus on methodology, development. But seems there is a lack of context with the neurosciences, and so the actually has a bigger plan in terms of RDoc, the research domain criteria. The idea is to integrate the information from the molecular cellular onto organ tissue and even the population level, right, across multiple domains. And imaging genomics seems only contributed to a part of the the analysis units. And while went to genomics conference like OHBM, right? So they are still focused on multi-omics, many other genomics factors, and imaging is only a small part of their interest. And so I still feel how to link the multimodal brain imaging with other omics data is a bigger challenge. In genomics, they have already developed many very good approaches, like a polygenic score prediction you mentioned of. Yeah, so I feel how to link the imaging with other modalities, including the cellular level imaging using the microscope. How to incorporate the latest developments in the single cell omics, the spatial transomics. So those are the the problems remaining to be answered. But there is a lack of data, the source of data, right? So for the same subject, same sample, can we have all those data sets from the other omics at a single cell level? Microbiome, metagenomics, proteomics, in addition to multiple brain imaging. So we have no access to those data sets, let alone building the models to link those different skills, and eventually how to use those multiple sources of data to answer the question proposed in terms of our talk. I think it's still a big challenge.
Yeah, I fully agree with you, Ping. I think that's a really challenging and I think Hongtu asked a super hard question. That's the reason I usually work a lot on imaging genetics. I even move away from that and kind of do a lot of like imaging with other health data. But I think we encounter a lot initially in terms of if we want to study the genetic by itself, we have a larger sample size. But if we insert imaging, then the sample size dramatically decrease. Particularly, we don't have a good size in terms of like disease sample with both imaging and genetics. So I feel with more data coming out, and there could be lots of potentials. I guess we can just wait and see whether we can just be part of that thing.
Yeah, I think thanks all the panel members, and I think for me, and because I know how people develop all these pre-trained models, large language models for CV problems and also for NLP problems. Okay, and usually we start with the concrete downstream tasks. Basically, the logic. It's the same thing for NIH. That's not the problem I want to solve. Okay. And then I go back to get all the data. Okay, that's the raw data. Okay. Then in order to train the models, this is the model I prepare for there. And then between I have to annotate all my data sets. Okay. If you think about imaging genetics, so the Paul mentioned the kernel questions. Then nowadays we get more raw data, okay? And then all these things between, maybe models, we don't need a very fancy models. These days, there's large language models, they just transform or whatever, modify a little bit here and there, right? That's basically this is the model part. But we still don't know in the between. Because for biomedical problems, it's much harder because in many cases, we don't know the ground truth. For NLP CV problems, for many problems, simple problem, for many problems, we know the ground truth in particular language, okay? And even for the NLP and those CVs, all the challenging questions, the people annotator don't know the ground truth. And then all the problems is stuck over there. Okay? Because if you don't know how to annotate the data, you don't know what's the model is, how to build the models. You don't know ground truth because you don't know how to evaluate it. Okay? That's in general, that's the opinion in my mind because I work in the tech for years. I know what it is, how do they build the models. Okay? That's basically, you start with the business questions, you get to the data, and you set up all these pipelines, then you need to annotate the data. If we don't have this, it goes nowhere. Because you don't know annotate. You don't know improve your data models. Because you know where to go. Then practically, when you really deploy the product to anywhere you want, you will cause a lot of troubles. The customer will complain. You've got a big consequence behind it. Okay, you see the point. I just wanted to emphasize the key point behind the research. It's not I tell the truth. Doesn't mean that's the we really need to move forward. Okay. Even though I tell you the key fact behind it. What's the key barriers behind all the research? I think Paul and I, have been working on this for enough time and know the key barriers. And another thing is the collaboration. I notice many people are trained to get a PhD in some specific areas. Some people from statistics, some people from mathematics, some people from the neuroscience, and psychiatry or the genetics. And then you come from different areas, right? But at the end of the day, when we work on all the problems we're trying to solve, it's not about a single field, okay? We are working as a team, okay? And together. And how do you see these collaborations? Maybe we'll start from you first.
Yeah, I definitely feel just particularly, I think I'm at a biostatistics department within a medical school, and it's lots of collaboration opportunity which I truly benefit a lot. And from my psychiatry and radiology collaborators, they actually have a lot of insights in terms of most cutting edge scientific question, but they don't know how to solve that analytically. And I think that's where we can play a role, rather than just be helping them and then play in their backyard. But maybe we can also just keep contributing our intellectual insights in terms of question to shape the question eventually. I think if from education or PhD perspective, I think joining those collaborations as early as possible definitely will benefit you to dive into the deep scientific question rather than just floating around towards methods.
That's a good comment. Yeah. So I benefit a lot from cross-disciplinary collaboration. Within the last 10 years, I work as a part of the bigger consortium among the 4 states, and so I learned a lot from my collaborators. And for example, our daily code, now the daily mind project. So we propose to use MEG, MRI, and multi-omics to study the brain development. So this is quite different from ABCD study, because ABCD does not have the MEG data. So when we have the brainstorming with my collaborators at the time when we put up our proposal, we thought this can distinguish us from ABCD study by using the high temporal resolution MEG data to study the brain dynamics at the nanoseconds resolution, while also incorporating the good resolution from good spectral resolution from MRI, in addition to genomics and epigenomics. We thought this is a good idea. If I do not talk to those collaborators, I wouldn't have a good understanding of those different modalities when studying the brain structures and functions. And I was trained actually from the math background, power, and in the past when I wrote a paper, if there is no equation or proof, that's not acceptable, right? So now I learned how to communicate people instead of using the mathematical language. And for my students, I told them, you have to force yourself, to read a science nature paper, to understand what are the true challenges in neuroscience, in brain research, rather than just methodology innovation, right? So we developed a model, but we are quite much inspired or motivated by the specific challenge in the data, especially in the context of neuroscience research. So I think the cross-disciplinary collaboration is really beneficial to people with a quantitative background. So I set up a lab in both our uptown campus and also the downtown medical center campus. So we can have daily interactions with basic biomedical researchers.
Yeah, all very good points. I mean, collaborative work is important, a lot of domain expertise, but something that I've noticed is how much I learned even about my own stuff through collaborators. I feel like sometimes people who have a little bit of data knowledge, I feel like they are way more creative than we are. Sometimes it feels like the fact that they are not bound by equations, they're not bound by expected questions from statistical reviewers and what statistical journalists expect from us, allows them to really be very creative, and I like it. I mean, sometimes they take shortcuts, so that's why collaboration, but I really got to learn a lot about my stuff too, in terms of trying to be a bit more creative from collaborators, not just domain expertise that they add to the topic.
That's a cool answer. And because this is a network meeting, I wanted to bring up the topology of collaboration. This is a very odd idea, but I'm curious what you think of it. So I've observed two topologies of collaboration. Let's have a think about each of them. So you have a network of collaborators, and there's edges in the graph. It's a social network. And there's information flow on the network, kind of like the brain, but people. So I've noticed two topologies of collaboration that are really effective. One is the relay race, where let's say someone invents stable diffusion in the mathematical field, in fact in the statistical thermodynamics field, and then the computer scientists implement generative AI for videos, and then the medical imaging community is doing generative AI for abnormality detection, and then the radiologist looks at your scan when you have a trauma, and that's like a relay race. So there were math people, computer scientists, data scientists, maybe neuroscientists somewhere along this. And the topology of collaboration is that the series of breakthroughs have a domino effect. And then in the end, the radiologist is using AI to look at your scan. Hopefully, when the end result is you get better, or there's less suffering. I don't know, maybe that's an objective. So that's collaboration topology one. The other one that's kind of cool is a hierarchical modular network. And this is, a lot of you are involved in collaborations a little bit like this. And mathematicians do this quite a bit. So there would basically be hubs, like an airline network. So I don't know how many of you flew into Charlotte or RDU. So there'd be a sort of hub. And maybe a hub is this meeting. I mean, there's a lot of people learning from each other at this meeting. So tomorrow, you'll have new information and new ideas, and you maybe have shared your ideas. So that's a hub. And then there's your plane home. So wherever you live, maybe you live here. Your plane home is a sort of long-range connection. And in the topology of networks, this is sometimes called a modular hierarchical network. And people argue they're in the brain as well. I don't know about that. But one of the things that I've noticed is that if we pay attention to the topology of the collaborative network that we're involved in, maybe we can improve it. So maybe we can all go to another conference. Or maybe you guys can give a talk on your work at a conference where people that don't know anything about this are. Or maybe, I don't know, the YouTube videos can be put online and someone who's stuck at home can watch them. So I'm sorry it's a slightly philosophical answer, but I think as we're network scientists, many people here are network scientists, you can think about information flow between yourself and your extended network of colleagues. And maybe how that actually can be updated, adapted, the edge strengths of the links can be built. Because part of what we're doing is social anyway, and you can analyze it as a sort of, a network as well.
Think about like the topology analysis. To that extent, I think I guess I can come from the perspective of like a young researcher growing up. I think this can be achieved through like personal level and maybe up to the team building level. Like I think most of us are trained as either mathematician or data science or engineering, this kind of so we are transferred a little bit towards the clinical settings. That's why maybe I can share some of my things. I don't think I'm the best example. I'm sort of pulled to the interdisciplinary collaborations. I think so personally, first level would be to get the things that you are interested most. So you probably want to focus on the for example, we don't want to understand the clinical fuzziness or the things. We just want a clear outcomes you can train on models. So that can make us sharpen our tools. And that's also created barriers of you want to jump out of your comfort zones and then to dealing with the difficulty with the problems. And I think to jump out first level, I think the social network is important. I think the size of the deep neural network, like for example, just coming out of the AIC, which is the largest conference in the Alzheimer's disease, I think it's, unless you need to identify the right group of people, otherwise the social network is too large for you to impact. And if you're stuck, if you're stuck within your own institution, then you don't have this level of interdisciplinary. So I think this size, sort of the size of level, is really force yourself to come out of the comfort zone and learn each other's. And I guess the other level is education. So you can't expect everyone ourselves to jump out of the comfort zones. So what education can be done, I guess, is when in the PI levels, when you have team building part. You probably want to, when I'm searching my postdocs, somehow I found myself more sort of leaning towards the team I see more diversity, where there's a neuroscientist, there's a clinicians, and I didn't realize that. I think in general, I feel those are the PIs who are deliberately creating these sort of a right-sized interdisciplinary environments that people can talk. And what I realized is the effect is that now I'm joining a medical school with the sort of engineering department centers inside that. And then this is even larger. So this is not a team level, it's an institutional level of collaboration. And when you are independent researchers at that levels, there's an actual real-world need that you have to get out, because independently you're not enough to answer. A lot of us are not enough to answer the real clinical question unless we are collaborating with the clinicians. It took me a while to understand what the clinicians is interesting about, because sometimes the question, their interest is not something that you can solve, or maybe sometimes you find it's not interesting enough, and so that's you need to dig into it, and then in the institutional levels that have different structures to cultivate the structures. I guess that's need the entire hierarchical of networks to make it happen. That's my thoughts. Thank you.
Thank you, and thanks to all the panel members. I just want to share my personal opinions, and I think all the comments are great. And I tell you the fact that, and I think if you really want to be a greater scientist, you have to be very hungry all the times, because you need to set your goals you want to achieve, but it's pretty high for you at the beginning. And if you really wanted to achieve that goals, most timely, you need to find someone to help you. Okay, that's how you move faster, right? And that's very important. You think about you spend years to get to the PhD degree. How many years? Okay, almost 10 years, right? If you really wanted to do something in other areas, do you think we really needed to start from scratch and learn everything from beginning? Almost impossible, right? If you have neuroscience questions, you have psychiatric questions, you ask the expertise, ask naive questions. Don't be shy on that, okay? That's the most important attitude, okay? If you don't know the math, some, specific theory, ask your mathematicians, and say, that's my question, I don't know anything, just tell me what's going on with this. We can collaborate, right? You always need to share something with your collaborators, okay? That's the most important point in this phase. To be a greater scientist, you need to really collaborate with other people closely and make a big impact in the field. Because when you think about it, even in the older mathematics, there's some older topics, they move very slow because there's no new challenging questions. Maybe the two challenges, nobody can solve it, okay? And you're stuck with it, okay? But if you change your different angles, you may see that you see a new world, and open a new world, and to solve the big challenging problems. There's so many challenging problems these days, and hanging around, just how do you solve it, okay? That's the most important part. Why is this interdisciplinary collaboration is so important for all of us. I think all the people here, senior people, I benefit from doing that. Okay? I think that's important also to tell the young generations. If you not just solving, I think that for PhD studies, you need to solve one problem. Going really deep, understand everything on that topic. And after your graduation, move out of your topic, okay? And look around, solve one problem, change it from a time. You spend these 5 years, I solve this, another 5 years, I solve that. The more, the time when you walk around, you see that you can do a lot of things, okay? I'm not regarding me as a statistician nowadays, and you can do all other things, right? That's the most important part, okay? I enjoyed the discussion, thank you. We have time for lunch.